Electrophoresis is a technique that separates molecules according to their shapes, net charges, and molecular weights using an electric field. When using electrophoresis for DNA analysis, we take our DNA sample and cut it into smaller fragments using enzymes called restriction endonucleases. These are complex proteins that recognize a certain short sequence of nucleotides and will cut the DNA wherever that sequence appears. In the DNA sequence shown, we see the nitrogenous bases of the DNA represented by the letters A, T, G, and C for adenine, thymine, guanine, and cytosine. A restriction enzyme will identify a certain sequence of around 6 bases, such as TGG, CCA. Anywhere this particular sequence appears, the enzyme will cut the DNA strand. Because each person has a different DNA sequence, restriction enzymes will cut each individual's DNA in different locations. This results in different sized fragments of DNA from each individual. Agarose is a gel made of polysaccharides that form a complex microscopic mesh. We will place the DNA fragments into this gel and pull the DNA through it using an electric current. DNA has a net negative charge, so it will be drawn to the positive pole when we apply the current. The DNA must navigate the spaces in the agarose gel. The smaller DNA fragments will have an easier time making it through these tight spaces in the mesh and move faster toward the positive charge. The larger pieces will move more slowly as they will have more difficulty squeezing through the gaps. The result will be lines on the gel with smaller DNA fragments farther away from the starting point and larger fragments closer to the beginning. Now we have a visual way of comparing one DNA sample to another, so we can tell if the two samples come from the same person or from two different individuals. This lab will identify a suspect in a crime. A similar method is used in crime labs as well as genetic testing labs screening for diseases, mutations, or other genetic abnormalities and similarities.